Hello, my name is Katharina Alamany. I'm doing my presentation on electrical pacemaker activity of the cardiac autorhythmic cell. So contractions of the heart, or heartbeats, are controlled by specialized cardiac muscle cells called pacemaker cells that directly control heart rate. This property is known as autorhythmicity. Neither smooth nor skeletal muscle can do this. Although cardiac muscle cannot be consciously controlled, meaning they are involuntary, the pacemaker cells respond to signals from the autonomic nervous system to speed up or slow down the heart rate. The pacemaker cells can also respond to various hormones that modulate the heart rate to control blood pressure. There are two major types of cardiac muscle cells: myocardial contractile cells and myocardial conducting cells. The myocardial contractile cells constitute the bulk of the cells, about 99%, in the atria and ventricles. Contractile cells conduct impulses and are responsible for contractions that pump blood through the body. The myocardial conducting cells are the autorhythmic cells that form the conduction system of the heart. Except for the Purkinje cells, they are generally much smaller than the contractile cells and have few of the myofibrils or filaments needed for contraction. Their function is similar in many respects to neurons, although they are specialized muscle cells. Myocardial conduction cells initiate and propagate the action potential that travel through the heart, muscles, and trigger the contraction that propel the blood. The wave of contractions that allows the heart to work as a unit, called a functional syncadium, begins with the pacemaker cells. This group of cells is self-excitable and able to depolarize the threshold and fire action potentials on their own. The feature called autorhythmicity, they do this at set intervals which determine heart rate. Because they are connected with gap junctions to surrounding muscle fibers, the specialized fiber of the heart's conduction system, the pacemaker cells, are able to transfer the depolarization to the other cardiac muscle fibers in a manner that allows the heart to contract in a coordinated manner. An experiment was done where the conclusion showed that if an embryonic heart cells are separated into a petri dish and kept alive, each is capable of generating its own electrical impulse, followed by contraction. When two independently beating embryonic cardiac muscle cells are placed together, though, the cells with the higher inherent rate sets the pace, and the impulse spreads from the faster to the slower cell to trigger a contraction. As more cells join together, the fastest cell continues to assume control of the rate. A fully developed adult heart maintains the capability of generating its own electrical impulse triggered by the fastest cells as part of the cardiac conduction system. The components of the cardiac conduction system include the sinoatrial node, the atrioventricular node, the atrioventricular bundle, the atrioventricular bundle branches, and the Purkinje cells. Action potentials are considerably different between cardiac conductive cells and cardiac contractile cells. While sodium and potassium play essential roles, calcium is also critical for both types of cells. Unlike skeletal muscle and neurons, cardiac conductive cells do not have a stable resting potential. Conductive cells contain a series of sodium ion channels that allow a normal and slow influx of sodium ions that causes the membrane potential to rise slowly from the initial value of 60 MV up to -40 MV. The resulting movement of sodium ions creates a spontaneous depolarization, or in other words, pre-potential depolarization, and brings the cell to threshold. At this point, voltage-gated calcium ions, the channels open, and the calcium enters the cell, forming the rising phase of the action potential and further depolarizing it at a more rapid rate until it reaches a value of approximately +5 MV. At this point, the calcium ion channels close and voltage-gated potassium channels open, allowing outflux of potassium resulting in repolarization. When the membrane potential reaches approximately 60 MV, the potassium channels close and the voltage-gated slow sodium channels open, and the pre-potential phase begins again. This phenomenon explains the autorhythmicity property of the cardiac muscle. There is a distinctly different electrical pattern involving the contractile cells. In this case, there is a rapid depolarization followed by a plateau phase and then repolarization. This phenomenon accounts for the long refractory period required for the cardiac muscle cells to pump blood effectively before they are capable of firing for a second time. These cardiac myocytes normally do not initiate their own electrical potential, although they are capable of doing so, but rather wait for an impulse to reach them. Contractile cells demonstrate a much more stable resting phase than conductive cells at approximately -80 MV for cells in the atria and -90 MV for cells in the ventricles. Since the cells reconnected by gap junctions, the action potential spreads from cell to cell by ion flow through the gap junctions. When stimulated by positive charge passing from neighboring cells, voltage-gated fast sodium channels rapidly open, beginning the positive feedback mechanism for depolarization. This rapid influx of positively charged ions raises the membrane potential to approximately +30 MV, at which point the sodium channels close. The rapid depolarization period typically lasts 3 to 5 milliseconds. Depolarization is followed by the plateau phase, in which membrane potential declines relatively slowly. This is due in large part to the opening of the slow calcium ions, allowing calcium to enter the cell, while few calcium channels are open, allowing calcium to exit the cell. The relatively long plateau phase lasts approximately 175 milliseconds. Once the membrane potential reaches approximately 0, the calcium channels close and the potassium channels open, allowing potassium to exit the cell. The repolarization lasts approximately 75 milliseconds. At this point, membrane potential drops until it reaches resting levels once more, and the cycle repeats. The entire event lasts between 250 and 300 milliseconds.
So to summarize everything in simple terms, we have the action potential, which is the electrical impulses in the heart, originated in specialized cardiac muscle cells called the autorhythmic cells. These cells are self-excitable, as we spoke about, able to generate an action potential without external stimulus by nerve cells. The autorhythmic cells serve as a pacemaker to initiate the cardiac cycle or the pumping cycle of the heart and provide a conduction system to coordinate the contractions of muscle cells throughout the heart. References are listed here.